Hello everyone, good afternoon. So welcome back to the last segment of the course. So we are at Unit 3. So I will be entirely dealing with Unit 3, and it consists of lecture on differentiation. Then we have exon path finding, synaptogenesis, neurotrophic factor signaling, all these. So today's lecture I'll be talking about is very kind of complicated lecture today, consists of a lot of genes. So I have mentioned in the slide, which slide you don't have to remember. So there are a lot of genes involved in differentiation. You have differentiation in the cortex, you have differentiation in the spinal cord. So I will be dealing with this today, differentiation. So we have talked about induction before, neural induction. So I'm not going to talk about induction today, but a little bit about determination and differentiation. So here there are some terms that I have definition I have included in the slide, so that at least you should know what is what. So induction is the process by which tissue is instructed to adopt a certain fate. Instructed to adopt a certain fate. That is we called as induction. Then determination is the commitment of a cell to a particular fate. It's already been committed from the beginning, like that is the commitment of a cell. We call it as determination. And the differentiation is the process by which a cell like neuron or glia cell attain all mature properties of the fate to which it has become committed. Like it has committed for morphology. There are set of genes which is required for express like to attain morphology or physiology or synaptic connectivity. So it attains all the mature properties of the fate. It is a post-mitotic neuron. It has all the mature properties. So in next slide, I'll be talking about different group of cells that that have all the properties that I mentioned before, like induction, or after induction, what happens, what is the next step. So these are some of the things that I have mentioned, like progenitor cells. So these are the dividing cells, okay, that respond to spatially or temporally varying cues that specify cell fate. So they have all these properties that can differentiate it to different cell types that we call as progenitor cells. Now you have another group of cells called as post-mitotic cells. These are mostly the newly born neuron, which have a specific transcription factor, okay, from the progenitor phase. And during differentiation, so those post-mitotic cells differentiated to different cell types depending upon the signal they get. So some of the cells differentiated to which helps in the biosynthesis of neurotransmitter, and some of these express genes required for the axonal growth or axonal guidance. So these are some of the common terms like used in this field, neural induction, patterning, and differentiation. I thought that I'll come up these terms so that you'll be able to understand what is what. So we will be talking about today neuronal differentiation. This is a complex process that integrates many signals and expression of many genes. You have expression of genes or signals, different genes or signals in the cortical for the development of the cortical layer, cortex. You have expression of different genes or a set of signals for the development of spinal cord. Okay, so because of this, and those are required to drive electrophysiological or morphological changes in the neurons. So that we will be talking about the neuronal differentiation, how the progenitor cells become post-mitotic cell and how it can differentiate to different cell types. So just to give you, it's not only about the genes are required for differentiation of different cell types. Sometimes some lineage and environmental cues and also required for to generate diverse neuronal cell types. So here this is a neighboring cells, and this is the cell membrane of the cell. Then this is the extracellular matrix. this is extracellular outside the cells. So this neighboring cell secretes a signal that is called signal molecule. It binds to the receptor, and now this receptor respond to this signal, and it generates a cascade of events. So the signaling cascade, which ultimately form the transcription factor, and it binds to the gene, and it transcribes the specific gene required to differentiate specific cell types. You have different varieties of cells like pyramidal neuron. You have different transcription factor for pyramidal neuron. You have Purkinje cells, different transcription factor. Okay, so this is just to show that it's not only the genes; it is also some these cues derived from other, cell types can also, differentiate to can also help in the differentiation of different cell types, neuronal cell types. So we will take classic example how different types of cells are produced in Drosophila. Mostly we're talking about the sensory vessels, that sensilla. So before going details into this structure, what is sensilla, what are the different cell types in the sensilla, I'll just want to orient where we are looking for and what is sensilla. So this is a Drosophila, adult Drosophila head, and it has eye. You can see that you have the eye here. Now you have antenna. So this is part showing here also. So this is the antenna, and you have this two small structure is called as maxillary pulp. Okay, so this is the antenna, and you have the sensilla present in the third segment of the antenna. So these are the spine-like structure. The spine-like structure, you can see these are the spine-like structure. These are the called sensilla, and the maxillary pulp is this is the structure of maxillary pulp. So maxillary pulp or the antenna contains the sensilla, and I want to show that how the sensilla is generated and what type of neuron basically required for generation of a sensilla in the antenna or in the maxillary pulp. Okay, so we'll go to the previous slide. So here, this is the structure of sensilla shown here in detail, but this is how it looks like. This is a diagrammatic representation, and this is how when it was imaged. So you have a sensory neuron that is going to the another structure called a socket, and you have a bacills like hair-like structure arises from the socket. This is called as bacills. So it consists of 4 cell types. So you have generation of you have the basal cells which produce the hair, and you have the socket cells which act as a kind of base for the bacills, and you have now you have the sensory neuron which is surrounded by sheath cells. So you have 4 cell types, and this is and each cell type contains different transcription factor to differentiate to different cells. So you have different transcription factor for basal cells, you have different for socket cells, different for sheath cells, and different for sensory neuron. Okay, clear? Okay, so and this is how the sensory neuron looks like in Drosophila developing larvae. So each Drosophila larva is highly segmented, and in each segment you can see that these are different varieties of sensory neuron, different diverse variety of sensory neuron present in this in the Drosophila larvae. And I will suggest don't remember this thing. What are the genes required for this sensory neuron? So this is only to show that sensory neuron present in larvae also. So so we will talk about how the different cell type is like different cell type. You have 4 different cell type that is produced in the sensilla. So this is a classic diagram showing the gastrulation of Drosophila embryo. So I talked about like embryogenesis in Drosophila in the very beginning of the class, like class 1 and class 3 and class 4. So lecture 3 and lecture 4, where I talk about after fusion of egg and sperm, they become zygote form blastula from gastrula, and you have different layer that is generated from the gastrula. You have generation of ectodome, neuroectodome, generation of mesodome, then generation of endodome. So this is a typical gastrula showing in Drosophila. So during generation of different genome layer, you have also initiation of a specific cell type that is called SOP. It is not statement of purpose. Don't confuse with statement of purpose. So it is called a sensory organ precursor cells, precursor cell division and differentiation. So SOP. So you have generation of SOP type cells in the gastrula. And in the next, I'll be talking about how SOP helps in the generation of different cell types in during formation of sensory vessels or sensilla. So here you have then you have the SOP, you have generation of ectodome, and you have generation of ventral nerve cord and mesodome. This is the different stage of the Drosophila gastrulation or embryogenesis. So we'll talk about more about SOP. So this is how the sensilla looks like. You have the sensory neuron, and this is the sheath cell. And you have the basal cell, and you have the socket cell, and here the basals arises from here, and you have the outside the cuticle. So this is the diagrammatic representation of a sensilla that I shown here. These are the sensilla, this spine-like structure. So the SOP, the sensory organ precursor cell division and differentiation, how it arises. So it arises from this during the gastrulation phase of Drosophila embryogenesis. So if you look at typical structure of like the SOP, so you have this is the kind of this is the dorsal side, this is the ventral side, you can say. So you have higher expression of notch expression in the ventral side, and you have higher expression of another transcription factor called as NOM in the dorsal side. So you have more ventral side notch, dorsal side you have NOM. Okay. So what happens is, the SOP kind of progenitor cells, you can say, the sensory organ precursor cells. So it divides to form two distinct cell types. One is SOP2A, SOP2B, SOP2A and SOP2B. So SOP2A has fed for differentiation to socket cell and basal cells, and SOP2B forms glial sheath cells and neuron. Okay, so don't remember this thing. Only kind of remember that you have the precursor, the progenitor cell, which can give different cell types because of expression of different transcription factor. Okay, so here I've written the higher notch activity promotes to form non-neuronal cell. When I was talking about notch delta signaling, I have mentioned that the cell which express delta produce more BHLH transcription factor, the basic helix loops helix transcription factor. In the last lecture, I think lecture number 5 or something like that. So that because of expression of BHLH transcription factor, you have formation of neuron. So that is because of more expression of delta. But if you have more expression of notch, it will produce non-neuronal cell. That's what you are seeing here. You have more expression of notch in these cell types. Okay. So this is a diagram showing how notch and NOM can regulate different cell types. So more expression of notch produce non-neuronal cell, and higher expression of NOM produce neuronal cell type, like glial cell, fed for the neuronal cell, like glial and neuronal cells. So here this is a diagram showing how NOM is distributed mostly in one daughter cell, so that one daughter cell can produce, one daughter cell for the SOP can produce neuronal cell type or the glial cell type. Okay, so here, so this daughter cell have higher concentration of NOM, and that's how you have when it forms two cells. So these cells contain higher concentration of NOM, and this cell do not have NOM. So that's why this cell become non-neuronal cell, and this cell become neuronal cell. So which is shown here. So this is a SOP. So when it divides it from two cells, and you can see that one cell contain NOM, other cell doesn't have NOM. That means this cell will form non-neuronal cell types, and this cell will form glial cells and neuronal cells. Okay. Any doubt? Yeah.
Sorry, which cells say have NOM?
Okay, so here if I show here, so this contain both, so NOM and notch. So here when it divides it from two cells, SOP2 and SOP2B. So this cell contain NOM. Okay, so that's what you are seeing. This cell contain NOM, so it become glia and neuron, and this cell doesn't have any NOM. It has notch. That's why it became non-neuronal cell types. Okay. So here in this diagram, I want to show that I am always interested to know if you mutate one specific type, what type of neuronal cell fate it will determine. So here, this is a wild type cell, SOP cell, contains both NOM and you have the notch. So you have notch activity, you have norm activity, and here because of norm activity, this cell produce sheath cell neuron, and because of notch activity, this cell produce socket and basal cells. So if I will give a scenario, if the notch is mutated, in the notch mutant, what happens? So notch mutant, what will happen is, so you don't have notch activity, so all the cells will become like SOP2B. So now this will forms sheath cell neuron, sheath cell neuron. So you will have two sheath cell, two neuron. Okay, because you don't have notch activity. That's why it's not producing this kind of cells. Now, if you have loss of function for the norm is mutated, what will happen? Now you have the notch activity, so that's why it forms socket cell, basal cell, socket cell, basal cells. Okay, so I purposely kind of put this slide because something I'm going to ask like this in the exam. Okay, so a researcher was doing this, and what he will find all this type of kind of analytical questions. So now, any question from the Drosophila segment, then before moving to the vertebrate segment. So I kind of thinking of touching both invertebrates like Drosophila and coming higher organism like mouse, human, and all these things, so that you will give an idea that how these things happens in the evolution. Okay. Any questions? Okay. So yeah, I know that this is a very complicated lecture because you have lots of content, lots of gene and all these things, but don't remember any genes. Only understand the concept, how different cell types become, how different expression of different transcription factor forms different cell types. So here, these are variety of, do not have to memorize anything, variety of transcription factor found in invertebrates and vertebrates required for the differentiation of genes. So we talked about this transcription factor, Ekite-Skute complex, the BLH-ALH transcription factor for the during the expression or differentiation of neuronal cell type in Drosophila. So this family, so the great thing that you are seeing, that is these transcription factor are present in the vertebrate, and blue is invertebrate transcription factor, and also there is some overlap also. So these are categorized into different family. You have E protein, you have a NATO family, oligo family like Ekite-Skute family. And in this family, you have this type of genes and which produce specific this type transcription factor. So you have in Ekite-Skute complex, you have the Ekite-Skute, you have lethalobscute, and in neurogenin family, you have the NG-N1, NG-N3. So these are the different types of transcription factor required for differentiation of different cell types. So so many transcription factor that specify cell identity, they mostly belongs to basic helix loop helix transcription factor, like in the Ekite-Skute gene in Drosophila, or homeodomain like Hox transcription factor. Okay, Hox classes. So this is the typical diagram showing the BHLH, the basic helix loops helix transcription factor. So this is the DNA. As I mentioned that the transcription factor, they bind to the DNA and induce the expression of gene. So so this is the DNA. This is the this is the transcription factor. You have two helix. You have helix 1. You have the helix 2. Then this is the loop, and you have the basic domain. That's why it is called as basic helix loop helix transcription factor. And most of the transcription factor that I talked about here, that belongs to this kind of. They have this kind of structure, and not all, but some of these, most of these. Okay. So then here I want to emphasize that which transcription factor, and you have various cell types, various neuron, a different diversity of a neuron present in the animal. You have the interneuron, you have the pyramidal neuron, different types of neuron, and different types of neuron have different kind of transcription factor, and different parts of the brain also express different types of transcription factor. So it's a complex thing, and just want to give overview. And this is not going to come in the exam. This a lot of things you don't have to memorize, but I just want to highlight some of the things like the mass transcription factor, and these are the mass transcription factor, mass 1, or you will talk about NG1, neurogenin 1 or 2. These are mostly the pan-neuronal. That makes the differentiate to only neuron. Okay, pan-neuronal, all the neurons. Okay, so So if you have the only expression of mass one, so it is a pan-neuronal genes. Mostly it deforms the neurons, the neuron. But when this transcription factor or this gene can interact with other gene, that forms other type of transcription factor. Okay, it can also interact with other determinants. Okay, other genes to form a other variety of transcription factor. So this is a kind of an interaction between one type of gene to other type of gene can produce another set of transcription factor. Okay, so itself it form the transcription factor for the pan-neuronal genes. Okay, but it can also interact with other genes to regulate the expression of other transcription factor. Okay, for the other neuron. So here these are the example. This is the forebrain, and just you can see that the MAS1 transcription factor, which is used to form the pan-neuronal for the neuronal transcription factor, it can also interact with other gene that is FOX2B, and this can also form this type of neuron, non-adrenergic neurons, which contains the dopamine beta hydroxylase. Okay, so it can form dopamine beta hydroxylase, which express in this type of neuron, because the MAS1 itself produce one specific type of transcription factor, but when interact with FOX2B, it forms DBH. So dopamine beta, it expresses dopamine beta hydroxylase, and basically, and this present in this type of neuron. Now you can see how different kind of diverse transcription factor are generated because of this kind of interaction. So this happens in sympathetic ganglia. So in the forebrain, again the MAS1 can interact with other determinant in the forebrain to form DLX2, which forms the GABAergic neuron. And in the spinal cord, you can see that in sensory ganglia, the NG1-2 forms this type of neuron, sensory neuron expressing BRN3A. And this the thing is that I want to highlight, you have the diverse type of transcription factor or diverse type of cell types are generated because of expression of different types of transcription factor, and that can happen because of interaction among the genes. Okay, so so that is what I want to highlight from the previous slide. Now that is all about in the brain that I talked about, but here I am talking about different types of transcription factor that also required for the development of spinal cord. So spinal cord has different type of transcription factor. So we know that sonic hedgehog expressed in the ventral in the ventral side of the neural tube, and and it goes down. You can see that the expression goes down in the dorsal side of the neural tube. So you have higher concentration of sonic hedgehog in the ventral side, and because of expression of ventral side, you can see that the class 2 transcription factor, which contain this variety of genes, dominate in the ventral side, but the class 1 transcription factor dominate in the dorsal side. So you can see that in the spinal cord also, you have different gradient of transcription factor, which determining the dorsoventral patterning. You can see that you have the different variety of progenitor cells in the spinal cord, and which can forms different type of post-mitotic neuron. So that's why, because of expression of different transcription factor, you have different cell types itself in the brain or in the spinal cord. Okay. So I will be talking about a little bit about different types, neuronal cell types in the spinal cord. So before that, I just want to highlight how the anatomy of spinal cord looks like, the cross section of spinal cord. So this is how it looks like. So you have a dorsal side and you have a ventral side, and you have the ventral horn. This is called as ventral horn, and you have the dorsal horn. Okay, dorsal horn and ventral horn, and you have the central canal. So these are the different anatomical feature of spinal cord. So in the next slide, I want to highlight what type of different types of neuron, mostly the interneuron, present in the spinal cord, that is innervating the spinal cord. So this is a typical diagrammatic representation of spinal cord, showing the dorsal horn and the ventral horn. In the previous slide, the dorsal horn and ventral horn. So you can see that it is in the gross anatomy shows that it consists of multiple layers. So you have layer 1, which is the marginal zone, the layer 1. This is a cross section of spinal cord. So you have substantia gelatinosa in the layer 2, you have layer 3, layer 4, layer 5, and layer 6. Layer 6 is the base of the dorsal horn, and layer 5 is the neck of the dorsal horn. So this is the dorsal horn. This is the dorsal horn. This is the ventral horn. Okay, so so you have different neuron innervates to the different layer, which expresses different types of transcription factor. Okay, so here you have, these are the interneuron that is innervating to the different layer of spinal cord. So you have three types of neuron that I have highlighted. So this neuron that takes receives signal from the nociceptor, which basically sense the temperature, noxious stimuli, and now you have this mechano receptor. That neuron is taking signal from the mechanoreceptor. Because of mechanical stimulus, it is going to through the dorsal root of ganglia. It is going to this again the layer 4, layer 3, and some part of the layer 5 also. You have other receptor. So that can also, that is called as proprioceptor. Okay, proprioceptor. Proprioceptor mostly that perceive the locomotion and movement and different action of different body parts. So this is anatomical feature showing that how different interneuron, and I'm going to talk about different transcription factor actually present in different interneuron. So it's just showing that the spinal cord, how different interneurons are present and how they are connected to the different layer of the spinal cord. So okay. So here again, this is showing the same thing. So we have the interneuron for that can sense the pressure, or that can sense the locomotion movement. So you can see that these are the interneurons. So different interneuron produce have different fed for different transcription factor. Okay, so here again, this is a very the complicated slide, but I want to make very simple. So here I'm showing what are the transcription factor expressed in progenitor cells. So you have a progenitor cells. Now you have the post-mitotic neuron, and that forms the differentiating cells. Okay, so these are the set of transcription factor present in the progenitor cells. So you can see that this is the spinal cord showing different expression of different genes. You can see that the DP1 progenitor cells, it has this transcription factor which is shown here, and even the P3 progenitor cell, you have the transcription factor like NKX6.1, 6.2. You have sub overlap of this green also. So it's showing that these are the different transcription factor expressed in different progenitor cell in the spinal cord. Okay, and here these are the different transcription factor expressed in the post-mitotic neuron in the spinal cord. And these sets of transcription factor they found in the sensory neuron, and these sets of transcription factor found in the motor neuron. Okay, so do not have to remember this slide. Just want to show that how complex it is, and the transcription factor and in different neurons. Okay, so and here I'm showing that it's the same thing. So here these are the progenitor cells, progenitor zone, then they become mitotic, and mitotic express the mitotic progenitor cells are mitotic, they become post-mitotic, expressing different variety of transcription factors. So here kind of highlighting one of the progenitor cell, that is motor neuron, progenitor motor neuron, and it expresses this transcription factor when it become post-mitotic, like HD9 and ISI12. So these are the cell types. It becomes motor neuron, and which have the acetylcholine neurotransmitter, and this motor neuron mostly innervate to the muscle. So post-mitotic transcription factors shape the differentiation of the neuron in each layer. Okay, so each layer has different types of transcription factor, different types of neuron. So how diverse it is to differentiate to different cell types. Okay, so this is just example of one of these I have shown. And in not only the transcription, different types of transcription factor that is present, present in during the development of the neuron, you have different sets of others, genes also expressed in this stage, which regulate, neurotransmission, cell adhesion molecule. Like you have this type of cell adhesion molecule which is presented here. You have different sets of gene also expressed that is required for the neurotransmission in addition to the transcription factor. Okay, so some of these are inhibitory neurons, some of these are excitatory neurons, which is color coded. The red is inhibitory and blue is excitatory. Okay, so just want to show different types of transcription factor. Now we'll come, and because of the expression of different types of transcription factor, you have generation of a different diversity of neuron. So you have generation of cortical pyramidal cells, you have generation of hippocampal, the CA3 neuron, and this is the Purkinje cells, and each of these cell types contain, have different transcription factor which can differentiate it to this type of cells. Okay, so we'll go some of these in the latter part of the discussion. And here showing this slide I have already shown multiple times. Here showing how complex is the cortex. You have different layer of cortex like layer 1, 2, 3, 4, 5, 6, and so the layer, these upper layer are formed later and the lower layer are formed earlier. So if you want to like label each layer, you have to, at specific stage you have to inject the radio labeled thymidine. So if you inject the earlier stage of the development, you can label the older layer like the lower layer, and if you inject in the latter stage the radio labeled thymidine, you can label the surface layer or the middle layer, something like that. So again, the different diverse kind of neuron that you are seeing here, the projection neuron, other neuronal cell types also have different variety of transcription factor, and all these types of you are seeing because of expression of a specific type of transcription factor. So obviously this neuron that is projecting to layer 3 have different type of transcription factor than this neuron which is present at layer 5. Okay, so we'll talk about some of these one or two of the transcription factor in the coming slide. So here you can see, because of expression of different types of transcription factor, you can see the different variety of neuron. This is a Purkinje neuron. You can see that different neurons are color coded. So how complex is the process? How complex is the development and the expression of different transcription factor generating different cell types? So here again we am going to the cortex, and here showing expression of different types of transcription factor can also happens in different layers. So here is the layer 1. The layer 1, layer 2, and layer 3 have these sets of transcription factor. So it forms the callosal neuron, and layer 5 have different these transcription factor, which mostly form these two varieties of neuron, that is corticospinal neuron. We'll be talking more about this for the layer 5, or the corticotectal neuron. Okay, and layer 6 have different transcription factor, which is shown here like corticothalamic neuron or callosal neuron. So and these are the transcription factors that are expressed in different layer. Okay, again, don't have to remember, but we'll talking more about this one corticospinal neuron for the layer 5 development. Okay, so here same thing, showing in different way, different types of transcription factor in different layer. So here this is the soft layer, then the layer 6, layer 5A, B5A, layer 4, layer 2, layer 3, and layer 1. So it has LHX transcription factor, which is showing that layer 1 has more expression of LHX, and less weaker expression. The light blue is weaker expression. You can see that the layer 5, layer 6, and layer at the soft plate has less expression of this transcription factor. So it is showing that how different diverse type of transcription factor express in different layer, and how much is the expression, and just showing the same thing in different way. So we'll talking about one of these transcription factor, that is FEZF2, and I'll be talking about FEZF2, its role in specifying layer 5 neurons. As I mentioned that we are going to talk about layer 5 and how the neurons are specified in the layer 5 because of the expression of this transcription factor, that is FEZF2 transcription factor. Okay, so FEZF2 transcription factor, I'll be talking more about this thing, and we will come to know how this particular transcription factor is actually helping in the development of layer 5. Okay, so don't remember any genes, but remember some of this highlight thing that I have mentioned here. So if you talk about this one, so in the layer 5, you can see that this is layer 5A and 5B. You can see that the stronger expression of this FEZF2, and you have a weaker expression on layer 6. You can see that it's not expressed that much in the layer 6, but you have more expression in the layer 5, the FEZF2. So we'll talk about this one, how FEZF2 transcription factor helping development of layer 5. So FEZF2 is a 4-brain embryonic zinc finger 2 transcription factor. So you have 6 type of this structure that forms this complex FEZF2. So showing one complex. So it's a zinc finger. It contains a zinc finger in the center. So you can see that the zinc finger, it has 2 cysteine residue and a 2 histidine residue, which is making coordinate bond to the zinc. So you can see that in the C terminus, this is showing only 1. So you have 6 like this that form this FEZF2 transcription factor. So showing the 1. So you have the alpha helix in the C terminus, and you have the 2 beta strand in the N terminus. And this alpha helix have two hydrogen, which is making coordinate bond to the zinc. Now you have the 2 cysteine residue again forming the coordinate bond to the zinc. So this is this is how the FEZF2 one subunit looks like. So you have 6 like this to make the transcription factor FEZF2. So this is a 3rd family of transcription factor we are focusing on. So before that, I talked about the BHLH, the basic helix loop helix transcription factor or homeodomain transcription factor. So this is the 3rd type of transcription factor we are going to deal with, and how this expression of this FEZF2 transcription factor actually regulating development of layer 5. So this is as I mentioned, this is called as 4-brain embryonic zinc finger 2 transcription factor, mostly expressed in the layer 5. This is a RNA in situ hybridization showing that it is mostly in the layer 5. And this is RNA in situ data. And this is again retrograde labeling. I talked about from the first lecture, retrograde, anterograde labeling. So you can do that with the DIL dye. This is again the postnatal postnatal day 14 during the development, the same embryo, the same brain. So you can see that this is highly expressed in the layer 5. So the FEZF2, which is shown here. So FEZF2 mostly expressed in the layer 5, and in the layer 5 you have special type of neuron that is called as corticospinal motor neuron. So if I'll go some of the previous slide. So you have, I was talking about the corticospinal CSMN, corticospinal motor neuron. So this FEZF2 mostly expressed in this type of neuron, the layer 5. Okay, so it expressed in this type of neuron, the motor neuron on the layer 5, and the axon of corticospinal motor neuron project to the spinal cord via corticospinal tract or pyramidal tract. So it is projecting to the spinal cord, from the layer 5, of the brain. So it is projecting to the spinal, corticospinal tract. Okay, so I'll show some of this data showing how defect in this type of transcription factor or FEZF2 transcription factor, how it is, how you have, poor development of in the brain. So that I want to highlight here. So this is a wild type brain, and this is stained with Nissl stain. Okay, in the past lecture I talked about different stain. So this is a Nissl stain, and Nissl stain basically helps in staining the cell body. So in the layer 5, you can see that CSMN neuron, that corticospinal motor neuron, which have the transcription factor FEZF2. You can see that you have bigger cell body. Here you have bigger cell body, which is in the arrow. In the arrow, you can see that, but in knockout mice, when you have loss of FEZF2, you can see that those bigger cell body are missing. So you have cell body missing in the layer 5 because of loss of FEZF2 in the layer 5, which is also shown here. You can see that this is a wild type mouse. So, and you have different marker for layer 5. So these are the protein which stands in the layer 5. So we have DIAP protein, you have a CRIM1, CLIM1, and this protein basically, these are the marker for the layer 5. You can see that those markers are present in the wild type mouse, either DIAP3 or CRIM1. These are present in the wild type mouse. But when you have loss of this transcription factor in the layer 5 of corticospinal motor neuron, you can see that these are gone. So here you can see that you don't have this expression of this marker or the protein in this knockout mice. Okay. So multiple layer 5 marker disappear in the knockout mice. So this is a RNA in situ hybridization data. So now another, so I saw that what are the different, proteins are missing in the knockout mice of FEZ-F2 transcription factor. When you have loss of FEZ-F2 transcription factor, I have shown that what are different other markers, other proteins are also missing. They're missing in this neuron. But here I want to show that different, some of the neurons are also missing when you have the mutation of FEZ-F2. So this is a wild type mouse. This is thalamus, and these are the neurons. You can see that in the wild type you have this, and this is done by anterograde labeling with the dye, DIL dye, or we have some data for the retrograde labeling also in the spinal cord. So you can see that you have this neuron present in this in the wild type mouse, which are missing in this FEZ-F2 knockout mice. So some of the neurons are missing, and also if you look at some of the projecting neuron in the spinal cord, this is called as a corticospinal motor neuron. This is present in this wild type mouse, and this corticospinal motor neuron are missing in FEZ-F2 transcription factor when there is a loss of FEZ-F2 transcription factor. Just remember that in the layer 5, you have special group of neuron called as corticospinal motor neuron, and that motor neuron expresses this transcription factor. And when you have loss of this transcription factor, you have loss of this motor neuron, which is shown here compared to that of control. Okay. So we'll I talked about loss of FEZ-F2. Now, what happens when you overexpress FEZ-F2 in the layer 5 or in the brain? What happens? So that can be done by under the specific promoter. So this is the cytomegalovirus promoter. This is a strong promoter which can express all the time. Okay. So they have cloned this the FEZ-F2, the gene under this promoter, to want to overexpress and see what happens when you express more of this transcription factor, what happens in the neuron or what happens in the developing brain. So here there are some marker for layer 5 and layer 6. So this is also I am going to highlight here the CTIP2 marker. Okay, so it is present in layer 5 and layer 6. So this is the GAP is cloned in the this is in this promoter, under the promoter to see this is used as a control. Now you have this FEZ-F2 which is cloned here. FEZ is cloned here under the you have this tag GAP tag. So they have injected this in the ETH 13.5 stage of the developing embryo, in the mouse embryo. So so you can see that. So the GAP is labeled. So this is FEZ-F2 present in the layer 5. So it is expressed in the layer 5. Okay. So you can see that this is this is expressed in the layer 5. So here this is the marker for this is the marker for the CTIP2. This is the marker for layer 5 or layer 6. So you can see that this is the layer 5. Okay. So CTIP2 express this is the marker for layer 5 or and layer 6, and they have injected this this construct, the FEZ-F2 construct in the 13.5 stage, which basically in that stage you have generation of a layer 4. Okay, so they injected that in the layer 4. So they have injected that in the layer 4, but and they sack this mouse in the layer in the 17.5. So you have that time you have a generation of a layer 5. So they have injected in the layer 4. Now they are analyzing in the later stage. Okay. So here I just don't want to focus it here. I just want to show that here so that it will be very clear from this diagram. So here this is a control GAP, and this is CTIP2. So this marks the layer 4, layer 5, and layer 6. So layer 6 is, or layer 5 is marked here, and this is a GAP, and they injected in layer 4. So when they injected in layer 4, so you can see that it is not co-localizing with layer 6 or layer 5 marker. Okay, because they injected in 13 point, where is that 13.5 stage, it marks only the layer 4. Okay, any confusion? So just forget about this one, but because this is more complicated, that's what I want to focus it here. So here they have cloned this control under the promoter of beta actin or cytomegalovirus promoter. Okay, and here this is they clone the GAP, and here they clone this FEZF2 that is expressed in the layer 5. Okay, so they injected that in the 13.5 stage, which actually in the lower which actually we can mark the layer 4 actually. So here you can see that since we have injected in the layer 4, so this is not co-localizing with layer 5 marker. So that's why you have separation of these two channels. You can see that the green channel is separated, the red channel is separated, because the red channel is for layer 5 and the green channel is for layer 4. Okay, because this is a GAP control. But when they injected this construct, the FEZF2, that is the marker for layer 5. So it should mark the layer 5 neuron, right? But when they overexpress that and injected in the layer 4, so it marks the layer 4. That's what I want to highlight. In the control, you do not have marking of layer 4 because it's not co-localizing with. So it is only marking the layer 4 and layer 5 separately. But here you have marking of layer 5 and layer 4 both. Layer 5 because you have this protein mostly localized in the layer 5. That's why you have co-localization with the red channel. Okay, so when you ectopically or overexpress the layer 5 FEZF2 in layer 4, it can also express in layer 4, and it can project neuron from layer 4 to other layer. That's what I want to highlight. Okay. So these are some of the summary of the transcription factor that is showing that FEZF2 is strongly expressed in layer 5, and FEZF2 is not expressed in layer 4. It is not expressed in layer 4, but when we are forcing to express in layer 4, it is expressing in layer 4, and it's projecting neuron which suppose not to project from layer 4 to other layer. The FEZF2 should project neuron from layer 5 to other layer. But when we are forcing the expression of FEZF2 in the layer 4, it is projecting from layer 4. Okay, that's what I want to highlight. So KO knockout, the knockout of FEZF2, you have lost in the layer 5 neuron that I have shown that. So here some more of the experiment they have done. So how FEZF2 regulate different genes? What is regulate? So what they have done here, so they have again cloned the GFP and the FEZF2 and injected in the mouse 14.5. They have labeled the mouse, and after 24 hours, they collected the brain. After 48 hours, they collected the brain, and they do the transcriptomic analysis. And they show that what are the different genes that is regulated by this transcription factor, which is present in the layer 5, that is FEZF2. So here showing that the variety of a transcription factor, some of the transcription factor are up-regulated, some are down-regulated because of the expression of FEZF2. So here you can see some of these transcription factor I have shown here, like LDB2. Then these are the different types of transcription factor. You can see that after 24 hours, compared to the control, you have more expression of FEZF2. You can see that in the layer 5, you have more expression of this type of this protein, because you have overexpressed this construct. You have more expression of this protein. So it can regulate, FEZF2 can regulate diverse genes, which are shown here. Some are up-regulated. So here you can see that this gene is down-regulated. This AKAP12 gene is down-regulated after 48 hours of injection of FEZF2. Okay, so it just want to show that many genes are directly up-regulated by FEZF2 because of binding to their promoter, and some genes are also down-regulated, which here this gene is down-regulated. Again, this gene is down-regulated, like TMEM163. You can see that when they inject FEZF2 at a P7 stage, you can see that this gene is down regulated after 48 hours of injection. Okay. So here I just want to highlight when they inject the FEZF2, this gene shows upregulation. You can see 24 after 48 hours. You can see that you have upregulation of this gene. So I'll be talking about some of this, one of the gene for how FEZF2 induces the expression. So here, this is how it looks like, the EPHB1 protein that is upregulated by this transcription factor FEZF2. So it is a multi-domain protein. So it has a heparin binding domain, it is a cysteine-rich domain, and the fibronectin domain. It is a kinase domain, it is a SAM domain, and the PDZ binding domain. So here you can see that these are the different developmental stages, and you can see that this protein, which is EPHB1, which is induced because of overexpression of FEZF2 transcription factor in the layer 5. So you can see that it is so higher expression, this EPHB1, so higher expression at 15.5 stage, and you can see that these are not there in other stages like P7 or later stage. So that means you have, when you overexpress FEZF2, it induces the expression of these genes, EPHB1 gene. So kind of showing that how transcription overexpression of particular transcription factor is regulating the expression of other, other gene product. So here you can see that in this wild type mice having the FEZF2, you have stronger expression of EPHB1, but when you have a mutant for FEZF2, you can see that these are lost. So here in the control, you can see that in the layer 5, you have FEZF2. When you have the FEZF2, you have expression of this protein, EPHB1. But when you have loss of this FEZF2, you can see that this protein is lost. Okay. So EPHB1 expression is lost in FEZF2 knockout mice. So in this knockout mice, you have loss of this protein. So that means FEZF2 is regulating the expression of, if you overexpress FEZF2, you have more expression of the EPHB1 protein. When you have knock out this FEZF2, you have loss of these things. So again, the FEZF2 can also helps in because of the FEZF2 and the protein EPHB1 protein can also regulate the the neuron, how it can regulate the axon guidance. So here it is showing here some of the 3 genotypes. So this is a very complicated thing that is represented here, but I want to make simple. So this is a control mice. So you have these genes, which is in the wild type condition. So you have EPHB1 gene plus, and here you have FEZF2. One copy is wild type, other copy for FEZF2 is lost by this construct, that is PLAP construct. So the PLAP construct basically is replacing FEZF2 coding sequence. So, and also it serves as an axonal marker. So the PLAP is replacing FEZF2. So here this mouse has one copy of FEZF2 and other copy is replaced with this marker. So this is served as a wild type mouse. So you can see that you have because of expression of these two, good amount of expression of these two protein, EPHB1 and FEZF2, you have projecting neuron to the corticospinal. You can see that the neuron is projected to the corticospinal tract. But when you have loss of EPHB1, the two copy is lost along with another copy for FEZF2. You can see that you have less projection. It is reduced compared to that of control to this region, the corticospinal region. But when you have loss of 2 copy of this FEZF2, because you are replacing this with this, so that FEZF2 will not be there, you can see that you are having loss of this, corticospinal projection. That suggests that the transcription factor FEZF2 or EPHB1, which is regulated by FEZF2, required for the axonal, axonal guidance, required for the projection of the neuron. Okay, so this is just to same thing. Just I am showing that FEZF2 and how it regulates different gene expression, including EPHB1 and you have different variety that is also mentioned here. Some are upregulated, some are downregulated. So you have downregulation of this one, which is shown in the graph. And this is the summary of the FEZF2, the transcription factor. So he is showing that some of these, it is required for the early and late the transcription of late genes for the corticospinal motor neuron. So it also inhibit the other group of neuron for the upper layer. It promote the V-glut neuron and other neurons. So this is a master transcription factor which regulate different neuronal cell types, development of neuronal cell types. So, and this is the last slide showing that similarly we have talked about different transcription factor and the regulation of the generation of a different neuron in the spinal cord. Here again, the diverse type of transcription factor can also including FEZF2, other type of transcription factor also required for the development of the neuron in different layer. So you have sub TB transcription factor for the layer 2, 3. So your FEZF2 for layer 5, layer 4, and mostly layer 4. So and also the layer 5. So you can see that layer 5, layer 4, and you have SOX5 and TBR1 in the layer 6. So you have this including the FEZF2. You have also the expression of other transcription factor which specifies or which differentiate neuron to the different layer of the cortex. Okay, so this is all about. If you have any questions, let me know. I know this is a very complicated lecture because it consists of different genes, but I just want to get a take-home message how different transcription factor regulate generation of different cell types in the developing brain.